hello there. We didn't see you.
We were just talking about this news article and it really got us thinking about genetics.
Oh yeah, the world of genetics is wonderful. Let us illustrate just how interesting it is to this up-and-coming technology we were talking about.
Oh, hello, I didn't hear you come in. So we've just been discussing about human-animal chimeras.
This article talks about how some scientists are combating the shortage of organ donors by looking at using animals to grow human organs. In Japan, guidelines were recently altered so that animal-human embryos can be implanted in a surrogate. Previous to this regulation change, scientists had experimented with chimera embryos, but terminated these a few weeks later. Pigs, rats, and even primates have been used as hosts for human organs. The bioethical concern is of cells migrating to non-target organs, especially the brain, resulting in animals with partially human cognition. Although scientists are proceeding with extreme caution, the idea of creating animal-human hybrids for the purpose of growing and harvesting human organs has many people concerned.
Originally, chimeras were mentioned in Greek mythology, that they were a mix of lion, goat, and dragon. Now today, chimeras, they're not our fire-breathing monsters that we have known. There's two ways to make a chimera. One way is mentioned in the article, fusing two species, and the other is naturally occurring. So naturally occurring chimeras result when in utero of one embryo absorbs another embryo. This leads to the first embryo gaining traits of the other, that they tend to have different hair types, two different colored eyes, or strange coat patterns, like this cat. There sometimes can be a chance that a male calico can be a chimera, but not quite all at the time.
How fascinating! Let's dive into this topic more in depth.
Cell differentiation is the process where a cell changes from one cell type to another and occurs as different genes are switched on and off. This allows adult somatic cells to be reprogrammed into pluripotent stem cells or iPS cells. iPS cells are undifferentiated cells reprogrammed to an embryonic-like state, which allows scientists to differentiate these cells into any cell type of the body.
Fun fact, we could all have our own pluripotent stem cell line.
There are two forms of mRNA processing that are involved in iPS cell expression: RNA interference and methylation. Genes can be silenced through the process of RNA interference. This is when small strands of RNA molecules, either microRNAs, miRNAs, or small interfering RNAs, siRNAs, are created from the cutting of double-stranded RNA by the enzyme dicer. They then pair with proteins to form an RNA-induced silencing complex, or RISC. These RISC complexes then pair with a part of the mRNA and affect it in a way that results in gene silencing. siRNA degrades mRNA before it can be translated, and miRNA incorrectly pairs with mRNA and inhibits translation.
The second form of mRNA processing is methylation of DNA. DNA methylation refers to the addition of methyl groups to the nucleotide bases. The addition of these methyl groups can activate or deactivate different genes and their expressions. Because of these changes can be passed to future generations, DNA methylation is maintained throughout DNA replication.
This study just touches on the surface of this topic.
Yeah, no kidding. There's much controversy surrounding this topic. It will be interesting to see how it evolves in the future.
Crossing over metaphase, I can recombine. Take a look, it's a GMO, a genetic sequence. I can go anywhere. Friends to know, an RNA to grow, a genetic sequence.